Welcome to another lecture of genetic engineering and its recent advancement. So we will continue our learning on CRISPR mechanism. Today we will look at it at a much detailed version and how we can have application of these CRISPR mechanism in order to develop a new recent advancement in case of biotechnology. So as we have previously seen, this CRISPR-Cas system works as just like a scissor in a system. So you have two strings and these two strings are intertwined with each other. For in case of DNA, they are intertwined with each other. So whatever foreign molecules enters to our body or any other organism, they tends to multiply themselves. In order to do that, what they will be requiring is they will be requiring to multiply their genomic DNA, and if somehow we can avoid this situation, we can avoid the increasing number of these viruses, bacteria or any other organism. So in 1987 in the University of Osaka in Japan, there was a study going on related to how the defense mechanism works in bacteria, and while they were studying this, they came across a short palindromic sequences. They term these sequences as clustered, regularly interspaced short palindromic repeats, and this leads to the acronym of CRISPR. Now they were studying in E. coli, and in E. coli they found there are certain sequences, they are continuously repeating themselves and they are palindrome to each other. These sequences are like just reverse copy of each other, just like in Hindi you have a word jahaz, whether you read it from the beginning or you read it from the backwards, it will termed as same jahaz. And in English word there is like race car, if you read it from the forward or backward it remains same, even though there are other sequences as well, but they are having a potential key role while playing in a defense mechanism of Escherichia coli. They are also found in, they have found the same mechanism in case of gram positive, gram negative and archaea. This creates a curiosity among the scientists why these sequences persist inside all these organisms, and these scientists were very curious to find out the importance of these particular repeats. And surprisingly, when they were studying Escherichia coli, they came across some kind of viruses. Now we know it's bacteriophage, and the bacteriophage feeds upon these Escherichia coli. These bacteriophage tends to climb on the surface of these Escherichia coli and insert their genomic DNA inside these Escherichia coli. Now what will happen is, when they have inserted their DNA, these DNA molecules tends to multiply and generate a new virion particle. But just as we as a human have a defense mechanism, both abbreviated as innate immunity and adaptive immunity, and you might have studied in the regular biotechnology courses, like in case of innate immunity we have macrophage, you have neutrophils, eosinophils, basophils, and in case of adaptive immunity we have B cell and T cells. These two work together in order to kill or in order to neutralize any foreign invaders. Similar to this, the bacteria also have the similar mechanism. Now if I talk about immunity, it will be incomplete without talking about the difference in case of primary response and in case of secondary response. For example, what vaccine does? So vaccines are nothing but the inactivated form of these viruses or these infections. So the primary response is lower, but if the organism came across the similar attack second time, the secondary response would be much more higher. At the same time, it required a very short span of time. Similar to these bacteria also acquired some sort of this adaptive immunity. So continuously these bacteria are being attacked by these viruses, or I would ratherly specify these as bacteriophage. So what happened is, if the bacteria is having some sort of defense mechanism for these different bacteriophage, what will happen is they needs to keep a tab or keep a memory of these different invaders. Now if you see or study the entire genome of bacteria, you will see a lot of spacer DNA. These spacer molecules are thought to be of junk molecule, but now we know due to recent advancement in case of genome sequencing that these spacer molecule are originated from mobile genetic element, also termed as MGEs, and they have a resemblance for the bacteriophage, transposome and lot of plasmid they have acquired from different other organisms. So just now, as I have stated, this spacer sequence belongs to a lot of different foreign DNA materials which have been recently discovered because of sequence analysis. It has raised a concern: could this be a defense mechanism for recognition of foreign DNA if they belongs to the foreign genetic material? So people have started to analyze in those times if they belongs to really a defense mechanism of these bacteria, for example as we are witnessing in case of E. coli. So now what happened in case of viral infection? When a viral load inserts its DNA inside any bacteria, what happened is you have this different arrays, or I would say different repeats of these small fragments of palindromes from different viruses. Now they form a array. If this array is called as CRISPR array. So whenever a foreign infection, a foreign viral load inserts its DNA inside any bacteria, it will recognize those small fragments which were thought to be already present in these viral DNA molecules, and along with different Cas protein, Cas1 and Cas2 protein, these molecules are recognized if they are already present in case of CRISPR array, as we can see from this figure. Now different infection or different viruses leads to similar activity, and whenever a new foreign material is being inserted, the cell will automatically recognize which part of DNA or which part of CRISPR array is responsible to recognition of that particular viral infection. So could this be a mechanism? Yes, people have thought about it and really start experimenting on it. Now you know how this happens. So once it is recognized, so this part is just recognition. What really happened after the recognition, that we are going to learn from this slide. As you can see, the bacteriophage has already inserted its DNA, and this DNA has a small fragment which is already present inside this CRISPR array. We are looking at example of a Streptococcus bacterium. Once this particular DNA is inserted, now your region from the CRISPR array where this particular region is already present is transcribed, and the transcription leads to RNA molecule called as pre-CRRNA. Now this pre-CRRNA is acted upon by a protein called as Cas9 protein. This is also termed as CRISPR associated nuclease protein, also short term Cas protein. So the category of Cas protein which is interacted by this pre-CRNA is Cas9 protein. Now this Cas9 protein act as nucleases, and we all know what nucleases does. This will cleave the pre-CRRNA. Now this particular, the category of protein which we are talking about, Cas9 protein, it's present in Streptococcus pyogenes. This is one of the very effective protein that is being studied so far and its effectiveness is being utilized for other purposes in biotechnology, we will see in further slides. Now, along with these Cas9 and pre-cRNA, there is another molecule called as tracer RNA. The time of cleavage, this tracer RNA also combined with the complex of pre-cRNA and Cas9 protein, as we can see from this figure. Now, this tracer RNA is complementary to the palindromic repeats. So, you can guess, as you have came across different palindromic sequence or different, you would see a different RNA molecule or different RNA viruses might have attacked it, and every different palindromes have a different different tracer RNA molecule associated with it. So, you have now in complex, you have pre-cRNA, tracer RNA and Cas9 protein. All these three work together and form as effector complex. So, these effector molecules are now responsible for the cleavage of these RNA molecules or DNA molecules, which DNA or RNA molecule which you have received from the foreign viruses. Now, these will act as a defense mechanism and immediately stop the propagation of this foreign viruses or the foreign RNA, foreign DNA of these viruses. So, now you have understood the whole part of it. Now, in this complex, you know the two different molecules, they are complementary to each other. For example, you have tracer RNA and you have pre-cRNA. They both molecules are responsible for recognition of these DNA molecules received from any viral infections. Together these three means foreign RNA, your Cas9 protein, tracer RNA as well as pre-cRNA form a complex molecule that is called as protospacer adjacent motif, also abbreviated as PAM. Now, once it is created, it will act as a nucleases and break down the entire, not the entire, but that particular section of virus DNA, and it will immediately neutralize the virus infection. As you can see from the human perspective or from the perspective where you have a defense mechanism against all these attacks, this will act as a secondary response because the bacteria is already having some sort of memory associated with these virus molecules. Now, how we can leverage these information for our own benefit? The two scientists, Dr. Jennifer Duoda and Emanuela Charpentier from 2012 studies these complex mechanism, and they studied it further for biotechnology purposes, and for their contribution they both have been awarded 2020 Nobel Prize in Chemistry. So, how we can use this? I will explain very briefly. So far we have studied in case of bacterial in case of viral infection, in case of bacteria, you have cRNA and Cas9 protein and tracer RNA. So, cRNA and tracer RNA, they both are different molecules, but we can actually add these two molecules and make a single molecule. So, we can add a linker and join together these two different molecules to form one single molecules that contain the cRNA as well as the tracer RNA, and we call it as sgRNA. These sgRNA are also called as single guided RNA, which we will be used to treat or to remove any unwanted infection from the system. So, suppose in a particular region you want to remove a particular section that is not desired. So, in a laboratory you can actually create this sgRNA having the srRNA particularly complementary to the region you would like to remove, and add a different and you can add a tracer RNA associated with this, and together they both act as a effector molecule along with the Cas protein, and you can simply remove that section you don't want to be there inside the genome. This is how this entire process is leveraged, and for our ease, all the gene knockouts and the other purposes, this method is used very effectively. So you have now understood how this entire mechanism works in a very simplistic way. People are using these methods very now and then to see if they can leverage this information and they can utilize this immense potential for their studies. Now what will happen is, once these in our experiments, once these gaps are created, means if the DNA molecule is cleaved, you will be remain with the cleaved DNA molecule. This cleaved DNA molecules are now the body's defense mechanism or the repair mechanism are initiated in order to join these two different flanked region again together. We will see how this works. So now we know how this cleavage or how these breaks are being inserted inside a genome, and we already know that body's own repair mechanism can repair these sequences and again join together with the help of some linkages or some ligases. So now we know the entire process, but as we were discussing the other day, the ethical considerations related to these experiments. So now this has opened an immense avenue for different experimentation that can being conducted using the CRISPR-Cas system. We can manipulate all these sequences, we can manipulate any sort of sequence, any sort of RNA, DNA, etc. We can even insert a new sequences using this CRISPR's method. So there should be a regulation or should be a guideline which should govern the entire process. As I was saying yesterday, there is a concept of green baby, or there is a concept of a baby which you can create as per you need. I would say it will be pretty cool if I have a baby with blue eyes, baby with 4 arms, and baby with 10 fingers. Just like in comic character you have superheroes, I would also want a baby that look like a superhero, but there is a major ethical concern associated with this. One should not play with these highly immense potential methods. Why, we will see in a bit. So it's like it has also generated a debate called as germline editing debate. One particularly continuous issue is the germline editing, which involves making any change to the DNA of embryo, eggs, or any sperm. This means that the genetic modification can be passed down for the future generation. Imagine, we have power to change the blueprint of future generation. While it could be used to prevent any genetic diseases, it also raises concerns about unintended consequences and playing with the nature. A notable example is the controversy surrounding the birth of world's first CRISPR edited babies in 2018, which sparked a global debate on the ethics of germline editing. Now, to address this concern, many countries have established regulations for the CRISPR technology. It's like creating a traffic rules to ensure the safety on the road. For instance, the US has a policy taken place to oversee and restrict certain uses of CRISPR, while countries like China have taken more protective approaches in pushing the boundaries of these research. The international organizations like World Health Organization, also abbreviated as WHO, have also been working on guidelines to harmonize the regulation across the borders. These regulations are crucial to strike a balance between scientific progress and ethical considerations. They are like guardians to keep the life on earth at its right path. In summary, the CRISPR ethical landscape is complex, with concern about unintended consequences in germline editing and global regulations. It's critical conversation that involves scientists, policymakers and public to ensure the responsible use of these powerful technologies. Now, I will briefly explain some of the very crucial applications of these powerful techniques. First, I will talk about applications in medicine. Now, we have seen the potential of CRISPR. We will explore how CRISPR is transforming the field of medicine. Think of CRISPR as superhero medicine needs to combat any genetic diseases. CRISPR is now treating genetic diseases. One of the most promising application of CRISPR treating any genetic diseases. Imagine having a tool that can correct the root cause of illness caused by a faulty gene. For example, sickle cell anemia is a genetic disease that affects millions and millions of people worldwide. CRISPRs have been used in clinical trials to edit the gene responsible for this disease, the potentially offering a cure. Yes, that's true. Similarly, in case of beta thalassemia, another genetic blood disorders, the CRISPR has shown promises in early stage clinical trials to correct the genetic mutations caused by these conditions. Now, the clinical trials and the success stories of these trials. Now the clinical trials are essential step in bringing CRISPR based treatment to the lab to the real patient. Think of them as testing ground to ensure the safety and effectiveness of these therapies. For example, in US, the first clinical trial using CRISPR to treat a genetic eye disorder called as the Leber congenital amaurosis showed the remarkable success. The patients who were once blinded now regained some of their vision, and it's one of the remarkable discoveries in the field of medicine. Now, the another notable success that involves the Emma, a young girl born with a rare genetic disorder called as diamond blackfen anemia. The CRISPR was used to correct the genetic mutation in her cells, potentially providing a lifetime cure. This success give hope to patients with previously incurable genetic diseases, showing that the CRISPR has the potential to transform the landscape of medicine. In summary, the CRISPR's application in medicine are groundbreaking, with the potential to cure genetic diseases that once are thought to be uncurable. The clinical trials and the success stories demonstrate the real world impact of these technology in improving the patient's life and their conditions. Now, let's explore the application of CRISPR in case of agricultural field. So, now CRISPR is revolutionizing the field of agriculture. Think of CRISPR as a magic wand for farmers, helping grow better crops and protect them from pests. Now, first let's talk about crop improvement and genetic modifications. Now, CRISPR has opened up the exciting possibilities in crop improvement and genetic modifications. Imagine if we could create crops that are more resilient, nutritious and better suited for our changing climate. For example, researchers have also used CRISPRs to develop a drought resistant crops. They are thrived in a region where they have limited water source. This could help to ensure the food security in those areas who are prone to drought conditions. Now, CRISPR has also been used to enhance the nutritional content of crops. Now, we already know about golden rice that was engineered using CRISPR. This contains the higher level of vitamin A, addressing a major deficiency in many developing countries. We will see another avenue in agriculture, that is CRISPR in a pest control. So, beyond improving the crops, the CRISPR is also being used in pest control. Think of it as a natural pesticide that specifically target harmful insects without harming any beneficial ones. The example of the use of CRISPR in developing gene drives, these are genetic modifications that are spread rapidly through the insect populations, reducing their ability to reproduce. It's like giving pest a genetic Trojan horse. For example, the researchers are exploring these gene drivers to combat diseases like malaria, modifying the mosquito to be less capable of spreading the diseases. This could have a significant impact in the public health. By harnessing the CRISPR in agriculture, we can create crop yield, we can increase the crop yield, reduce the need for harmful pesticides, and work towards more sustainable and resilient food supply. In summary, the CRISPR's application in agriculture are diverse from the improving crop resilience and innovative pest control methods. These advancements are crucial in addressing the global food challenge and protecting our environment. I would say, if I have an example of a particular insect which continuously affect a crop in a particular region, I would certainly find out the chemical or the potential target which can repel these pests. I would recognize a exact molecule and its associated protein, and then I would do the reverse engineering and find out the exact gene responsible for this kind of action. I would like to insert these inside my crop and then try to avoid the attack of these pests. In the same way, I would also not play with the balance of the nature to just simply remove all these pests in that particular crop. So, in this way, I would be needing a lot of checks and balances about how these genes affect the natural physiology of my crop. If it does, it will lead to a very bad situation. So, as you might see, there are a lot of checks and balances that needs to be required in order to keep this technology under the bucket. These have immense potential in all this field, specifically in case of agriculture. Now, as you see, as the population is increasing, the demand of having more and more food supply is also increasing. This had led to a situation where we are continuously using the chemical fertilizers or other different harmful chemicals. They can potentially insert themselves inside a human or animal system. In previously, it is known that a lot of, in case of different birds or in case of different insects, they have also been inserted inside these insects, and somewhat it has inserted inside our food chain. We would like to avoid this situation, because ultimately it is also, it depends upon the predictive science whether the effect is minimal or you can actually witness the effect after a certain number of duration. We also wish to know if these changes that you are witnessing in this particular generation, they can be transferred to the next generation or not. If it does, you have to keep a checks and balances between all these situations. So, with this, we will continue our studying of these potential areas in biotechnology or other field in our next lecture. We will also see some of the very specific case studies associated with this powerful tool called as CRISPR in our next lecture. Until then, I wish you happy learning. Thank you very much.